Hi, my name is Osnat Yanai and I'm the Tissue Culture Lab Manager, and I'm here to tell you a little bit what we're doing here in the lab. So Danziger has a complex of 3 labs: the Diagnostic, Research and the Tissue Culture Production Lab. In the Tissue Culture Lab, we have 2 responsibilities. The first one is keeping a clean, vegetative, disease-free genetic stock of all Danziger varieties. The second responsibility is the production. We multiply these plants, the commercial varieties of Danziger, in order to send them and to establish the mother stock plant in different sites of Danziger around the world, such as in Colombia, Ecuador, Kenya, and Guatemala. The tissue culture procedure includes 4 stages: introduction, multiplication, rooting, and hardening. Let's take the Capella Hello Yellow, our new introduction for 2022, as an example. We are starting the procedure with the introduction by using 50 cuttings that we are ordering from the greenhouse. We disinfect them using bleach solution that contain 1.5 sodium hypochlorite, and then we are moving to work inside the laminar. Here you can see Galena that works inside the sterile environment, in which the air is coming from the outside through a filter and inside. After the disinfection, she washed them off using sterile water, dried them, and getting them inside the tube. In the tube we have a media that contains agar that stabilizes the plant inside the tube, and all the minerals, nutrients, and vitamins that the plant needs in order to grow. After 2 to 3 weeks, we are checking the plants to see that there isn't any unwanted bacteria or fungi that developed, and then we can move to the next stage. The next stage is the multiplication. In order to make from one plant lots of plants, we cut and separate between the parts of the plants. We are taking the axillary meristem and the shoots and placing them in a new media, the same substance mix as before. Each variety has different multiplication rate, and we are taking it into account when we need to time for the timelines of the shipments. After this stage, we need the plants to get into roots in order to transfer to the soil. So sometimes we are adding auxin, which is a growth hormone, to the media in order to accelerate this stage. In this stage, we are sending the plants, the boxes, to different Danziger sites around the world, as I mentioned before. And then they are doing the final stage of hardening, in which they are taking the rooted plants from the box into the soil. This should be done very gradually because the plant is coming out from a box with 100% humidity. So they planted them into the soil and covered them using a plastic cover for something like 10 days to see that the plants get new leaves and that the roots become much bigger inside the soil. After that, the plants can use as a mother stock, and from that plants, cuttings are sent to different customers all around the world. This was a short review of what we are doing here in the lab. I hope to see you in person next year. Thank you.